This recording is going to look at the role of the small intestine in digestion, and kind of as an overview is the small intestine is broken up into three regions: the duodenum, which is closest to the stomach, then followed by the jejunum, and then finally the ileum. Now, kind of just broad, and it's actually going to be explained by kind of the structure of each part of the small intestine. Is in the duodenum is receiving the chyme from the stomach, secretions from the gallbladder, the liver, and the pancreas. And in the duodenum, we're primarily trying to get the contents from the stomach neutralized so the pH is proper for digestion. There's not a lot of digestion that takes place in the duodenum. But the duodenum does have a lot of glands we call Brunner's glands, which help to protect the mucosal wall from the acidic chyme that is coming from the stomach. Now, the jejunum, this is where the majority of digestion and absorption of nutrients takes place. And then we have the ileum. Now, I'm going to do it now because I know I'm going to forget it later. A couple major things that take place in the ileum. One of them is the absorption of vitamin B12. Takes place here. Intrinsic factor, which was secreted by the stomach, binds to vitamin B12 from the food that you've eaten, and actually at the ileum it aids in the absorption of vitamin B12. Another thing that takes place in the ileum is we have recycling of bile salts. So bile salts we call it, there's a term called enterohepatic circulation of bile. And so some of the bile salts are taken back up and reused to help with the production of bile in the liver. So those are two big things that actually take place in the ileum. But let's kind of look at that structure of the small intestinal wall. So the mucosa in the small intestine, we may see these circular folds or folds of the mucosa called the plica circularis. And on top of the folds, you'll see these villi. Both the plica circularis and the villi help to increase surface area for absorption and secretion, and because the small intestine is so important in that. Now here's actually a microscope slide so you can see. Here's the plica, and then you still see the villi here on it. And the villi, and on a light microscopy, it's very hard to see, but you see here that's just looks just like a little fuzz area. It's the brush border. That's actually microvilli. To really see the distinct little microvilli like this, you'll need an electron microscope. But the microvilli also increased surface area for absorption and secretion. In the brush border, those cells that we find in there too also are producing various secretions, various enzymes and other things, and even some hormones. Notice you have some goblet cells here too, and the goblet cells produce mucus, and that mucus does protect the mucosal lining from what we call excoriation. So it doesn't kind of get worn away, and mucus actually prevents that all the way throughout the GI tract. It also just protects the lining from being digested itself. You may also see in the intestine some of the lacteals, and that helps with the absorption of dietary fat too. Now in lab, I've mentioned that the small intestine, the wall, is different depending on where you are. In the duodenum, they have plica and they have villi, but they're more leaf-like. The villi are very long and finger-like in the jejunum, and they have extensive plica. And when you look at the ileum, the villi are much shorter than you see in the jejunum. And so having those really long finger-like processes in the villi, I should say, in the jejunum helps with the digestion and absorption that you have there, because that's where the majority of digestion and absorption takes place, is in the jejunum. Now, the intestine does contribute secretions for digestion. So the brush border cells secrete peptidases, and peptidases digest peptides. So peptides are short chains of amino acids. These are breakdown products from proteins. So you start digestion of proteins in the stomach using pepsin. And you also have some more digestion of proteins using the enzymes from the pancreas, but you're not going to get complete digestion of those proteins. And so you may get to levels of the peptides. Well, you can't absorb peptides, you absorb amino acids. So peptidases digest peptides into amino acids, and then that's what we'll be able to absorb. The small intestine also secretes enzymes that break down disaccharides. They're referred to as disaccharidases. Sucrase is an enzyme that digests sucrose, and sucrose is a disaccharide composed of two monosaccharides. So with the digestion by sucrase, you get fructose plus glucose. So you may get sucrose. It's from it's from sugar. So that's needed to digest sucrose. Maltase digests maltose, and maltose is broken down into two glucoses. And you can get maltose like in things like beer. Maltose can also be the result of the action of amylase. So amylase digested starches, and starches get down to the level of maltose. Now you need maltase to finish everything off. Lactase digests your milk sugar, called lactose, and when you break that down, you get galactose and glucose. So while I'm here, I want to, what I'll do is while I'm at the end, we're going to go through the major macromolecules, and we're going to say where these molecules start their digestion, where they're completed, and list the different enzymes involved and where those enzymes come from. So start thinking about that, because we're going to do that at the very end. The the small intestine also secretes lipases. We saw lipases coming from the pancreas too, but lipases break down triglycerides, fats, into fatty acids and glycerol. Enterokinase, we've seen that previously in a recording having to do with the pancreatic and certain pancreatic enzymes. Enterokinase is an enzyme that's found on the brush border that converts trypsinogen to the active proteinase trypsin. So trypsinogen came from the pancreas, and it has to be activated into trypsin. Trypsin then activates all the rest of the enzymes, proteolytic enzymes, released by the pancreas, and that helps with the digestion of proteins. Enterokinase is needed for the activation of trypsinogen, and that's produced by the small intestine. There's actually a multitude of hormones, but two hormones produced by the intestine are cholecystokinin and secretin. So let's kind of review some of what cholecystokinin does. So cholecystokinin, one of the things it does is it causes, in no particular order, is it causes contraction of the gallbladder, causes relaxation of the sphincter of Oddi, which is the hepatopancreatic sphincter. So we can have the contents from the common bile duct as well as the pancreatic duct empty into the duodenum. It stimulates secretion of pancreatic enzymes from the pancreas. It also causes contraction of the pyloric sphincter. So the pyloric sphincter controls the emptying of your stomach. So if you think about it, cholecystokinin, its main potent stimulus for its secretion is the presence of fat and protein in entering the duodenum. We need the bile from the gallbladder. We need pancreatic enzymes to help with the digestion of those things. We want to slow the rate of gastric emptying because I need time to get everything ready so I can have proper digestion of those things. So it slows the rate of gastric emptying. If you think about it, if you have a meal that's a little bit higher in fat, a little higher in protein, you stay fuller longer. And because that's true. It really is. At least in my instance. I get to do, personal experience. Now, the secretin is what it does is it stimulates bicarbonate secretion from the ductal cells of the pancreas. Big stimulus for its secretion is the acid. The more acidic the kind, the more secretin that's released, and that releases more bicarbonate to help to neutralize that. So it has other actions, but that's the main action. Some things that coordinate secretion as well as absorption of the nutrients. So once we get the digestion, complete digestion, is you do have local reflexes, just like the presence of things along the wall will stimulate secretion of things. You have the role of the enteric nervous system. So the enteric nervous system has two components. So remember you have let's do this one. You have the myenteric plexus and you have the submucosal plexus. So the submucosal plexus is located in the right in the submucosal area, and that's going to be responsible for controlling secretions and blood flow. If you think about it, is you need secretions for substances to help with digestion, and you need to have blood flow to be able to carry all the nutrients away. Myenteric plexus is more important with controlling motility, but enteric reflexes would be stimulated by the presence of certain substances in the what's the word I want? The food stuff that's in the small intestine, presence of acid or protein or fats. They stimulate, or just mechanical or things, the tactile touching of the cells stimulate these reflexes. Say, okay, I got food here. I need to have more secretion. So you have enteric reflexes that are involved. The autonomic nervous system may also be involved, and specifically the parasympathetic. So this will be important when you're digesting the food, is the parasympathetic division may play a role too. Sympathetic is inhibitory. So parasympathetic stimulates secretions, also would help with motility in that small intestine. The movements, so speaking of motility, movements that we associate with the small intestine includes that forward propulsive movements is peristalsis. You have the segmentation, where if I kind of break this up into, it's not the prettiest picture, so you're kind of breaking the material up into little segments. That helps with mixing and helps with digestion. And we've mentioned this in a previous recording. Now we do have some reflexes associated with the intestine. Gastroenteric reflexes, so the presence of foodstuff in the stomach, trigger motility in the small intestine. So I want you to think about it. Food hits the stomach, it's saying something's going to be coming along pretty soon. Start moving things along that may be in the small intestine. So we have gastroenteric reflexes that are involved. Gastroileal reflex. This is where presence of food in the stomach trigger relaxation of the ileocecal valve, which is the valve that controls the movement of material from the ileum into the large intestine. So that will stimulate relaxation of it, again helping to move things along to free up room, because we're going to have more stuff coming in. Now what I want to do, so I told you I wanted to do, is I want to go through certain macromolecules and go through where the digestion begins, where it's ended and completed, and factors that are involved in it. So let's start with starch. So starch digestion begins in your mouth because saliva has amylase. But amylase, pretty much the farthest we can go down is we're going to be able to get to maltose. We can't absorb maltose. So we're going to need to finish that off. So we start digesting the starch in the mouth using amylase. It hits the stomach. Once that pH of the stomach drops too low, that amylase is not going to be working. So we're not going to be able to finish up digestion of starch in the stomach. So just not much going to happen there. You hit the intestine. And in the intestine, you're going to have amylase from the pancreas. So pancreatic amylase gets added to the small intestine. That again, any starch that was left, we're going to again break it down to the smallest I can get is maltose. And it's actually, we should put amylase is digesting starch down to the maltose. But maltose, I said, we can't absorb it. Maltose has to be converted to 2 glucose molecules using maltase. And maltase came from the small intestine. So you have secretions from the salivary glands, the pancreas, and the small intestine. So you have amylase and maltase that help with the complete digestion of your starches. It begins in your mouth, it ends in your small intestine. You'll then absorb the glucose in the intestine. So that's starch. Now let's do fat. So fat, I always regret saying this, but there is such a thing as lingual lipase, which is in your mouth, but you can't digest the fat in the mouth, so I'll just ignore that. So fat digestion begins and ends in the small intestine. So write that down. It begins and ends in the small intestine. So what do we need for the proper digestion of fat? You need lipases. Lipase comes from the pancreas and the small intestine. But you also need the help of bile. bile makes the lipases work better. So bile comes from the gallbladder, and the liver. It's actually synthesized in the liver, but stored and concentrated in the gallbladder. So once you get fat broken down to all its constituent parts, then we'll have the absorption there in the small intestine. So fat is digested and absorbed. All of it's done in the small intestine. Okay, let's move to protein. That's the last one, or it's not the last one, because I'm going to do this because I'm going to forget. So I want to do disaccharides too. So I'm just going to do that so I remember. Okay, so proteins. Protein digestion begins in your stomach. And when I say digestion, I'm specifically referring to chemical digestion, where we're breaking bonds. You can mechanically break things down into smaller parts, but you're not breaking the bonds in the food. So protein digestion begins in your stomach because pepsin. So pepsin was produced by the chief cells. It's actually released as pepsinogen, but it has to be activated to pepsin using hydrochloric acid. So pepsin helps with digestion. We will then need enzymes from the pancreas. So we're going to have pancreatic enzymes that will be needed. And those enzymes will be activated in the stomach. And so they were trypsin. I'm just doing the activated ones. Trypsin, chymotrypsin, carboxypeptidase, elastase. So these will break down the proteins to peptides. Then we'll need peptidases released by the small intestine to finish it off. So what we'll get at the end are amino acids. So we need secretions from the stomach, the pancreas, and the small intestine to get complete digestion of proteins. It begins in the stomach, it ends in the small intestine. Now what about disaccharides? So I say maybe drink a glass of milk. If I drank the glass of milk, I'm not going to digest it in my mouth. I'm not going to digest it in my stomach. Lactose is the digestion of lactose begins and ends in the small intestine. So begins and ends in the small intestine because it uses lactase from the small intestine to convert lactose into galactose and glucose. Sucrose, sugar. I've been eating a lot of candy lately. A lot of sucrose in that. Sucrose digestion begins and ends in the small intestine because you have the enzyme sucrase in the small intestine. Well, what about maltose? Well, maltose digestion begins and ends in the small intestine because you have the enzyme maltase. So these are the major things I want to go through because I want you to realize kind of what components of the digestive tract contribute to the digestion of various macromolecules. Where does it begin? Where does it end? Okay, so let's leave it here. Next recording, we're going to talk about large intestine.